Hello friends, welcome back to my YouTube channel Astin Parasali. So in this video we are going to see list of questions that was challenged by student, and we are going to see about the outcome of challenging responses, everything with regarding to shift 2. First question is 703600. NTS mentioned option number 2 is correct. According to me also, I am agreeing with the answer that was given by NTA, but many student has challenged this particular question. Okay, I had also added up the image that I had taken from Ty's book, page number 600, recent edition, 6th edition. So if you are challenged this particular question, kindly provide me the reason why you are challenged this particular question. Next is question ID 703597, for which the correct answer is option number 3. That is, they had given 5 statements. They also asked us to pick up the correct statement. So only option B and option E alone is correct, because if you check option A, it is reason, but here they had mentioned hemoattraction is mediated by binding, like that they had given. Okay, so if you check option C, the capacitated sperm, but here they are asking about sea urchin, the sperm, that is mammalian sperm alone, will be going through a process called capacitation. So option C is also incorrect. Next is option number D, that is acrosomal protein mediating the critical species specific binding event is bind in it, but here they had given reset. So they had ulta the option A and D. So the correct statement is option number 3. I am agreeing with the answer that they had given provided by NTA. Next is 703544. They have given statements of photosynthesis and they are asked us to pick up not correct statement. And the answer that officially given by NTA is 4, because if you check option number 1, light reaction takes place in thylakoid, absolutely correct. ATP and NADPH are produced in thylakoid membrane, of course it is correct, because ATP and NADPH are produced during light-driven reaction which will be taking place in thylakoid membrane, and both are consumed in carbon reaction which is taking place in stroma. So option 2 is also absolutely correct, but here they are asking which statement is not correct. Okay, third statement is also correct, but the fourth statement is wrong. NAPDH is produced during carbon reaction by enzymes present in stroma, but it is produced during light reaction. That might be the one reason, but many students has challenged this particular question. Okay, the reason I am predicting is that some are telling like statement 2 is also not correct, because ATP is produced in stroma, like that you are telling. Some student, if you notice, if you take statement number 4, the answer is typed wrong. It is NADPH, but here they had given NAPDH. Okay, so there is a spelling error, but we might not know whether CSR will be taking this particular thing into account, because I had noticed that particular thing. Okay, but statement number 4 is not correct. Okay. Next is theory of island biogeography has synthesized into the theory the following concepts except. So I am agreeing with the NTA answer, because NTA just mentioned speciation. Speciation has nothing to do with island biogeography, because there is a difference between speciation and number of species. So the correct answer is option number 4. I am agreeing with NTA answer. So please do tell me if you had find some other reason. Next is question ID 703579, that is with regarding to nuclear transport. Okay, sorry, it is about cytoskeleton dependent motor protein with regarding to movement of cellular organelle in animal cell, for which the correct answer is dynein. So at the nuclear envelope, the dynein will be moving along the microtubule towards the N minus A. So minus N will be present in the centrosome. So correct answer is dynein is the correct answer. So you can refer this particular article. So please do tell me if you want the link of this particular article. So based on the diagram, the cytoskeleton motor protein that is critical for the movement of organelle is dynein. Next is this is the most viral question, that is 703550, where reduction in frequency of heterozygous genotype with a concomitant increase in frequency. Sorry if I pronounce them wrong. Increase in frequency of homozygous genotype in context of random mating. Okay, so for which they had mentioned option 2, intense inbreeding is correct, but here they are not speaking about non-random mating, because intense inbreeding will be coming under non-random mating. Okay, here they are speaking about random mating. So many students have challenged this particular question. I couldn't able to get a correct research article stating this particular statement. So if you have already holding some other statement, please do share me in my mail ID. It will be really helpful for many students. Many students are demanding option 1, that is genetic drift is correct. Intense inbreeding will be correct if here they have given non-random mating, but here the question they are asked about random mating. Okay. Next, some students are challenged about this particular question, but according to me, it's a correct statement. Okay, so it is option number 1, that is glucosinolates. Okay, next is 703523. Again, some students challenge this particular question, but we all know it is the easiest ever question as asked in shift 2. Okay, it is correct answer is lysine. Next, some students challenge 703601. I don't know what is the reason for them to challenge this particular thing. Next is 703537, which means in avian embryo, that is blastocele like fluid filled cavity will be forming between epiblast and hypoblast. Last time itself, when I was discussing unofficial answer key itself, I have provided you a diagram and all. Next is 703585, for which they just mentioned following statement were about post-transcriptional processing, for which the correct statement is option number 4, that is A statement alone is correct, but according to me option D is also correct. Okay, instead of giving fourth option, for me the correct option is option number 1, which includes statement number A and statement number D, because capping of eukaryotic mRNA occur exclusively in nucleus of the cell. Okay, so here you can able to, in this article you can able to find, okay, rest of the things are incorrect. Option B is incorrect. It is not with regarding to removal of group 2 intron. It is a completely different concept. At the same time, transsplicing will be occurring between two exons. Okay, it is between two genome, two genetic segment. But here they had given 3 or more. Transsplicing generally occurring between 2 exons. Next is some students challenge question ID 703570. This is the correct statement. The answer that was given by NTA is option number 1. I'm agreeing with the NTA answer. Next, again some students challenge 703567. So which of the following statement is not true with regarding to continuous culture batch fermentation? So that is option number 3. Risk of contamination will be high in continuous culture, because batch fermentation it will be conducted in a closed environment. Okay. So some students challenge. I don't know the reason for them to challenge this particular question. So please do tell me if you are challenged this particular question means, kindly do tell me, friends. So next is unikaryotic gene DNA sequence that define promoter occur in either region upstream of transcription site or within a transcript region also. So I'm agreeing with the NTA answer, but some students challenge this particular question. Next is the important question. 703528, that is which one of the following condition associated with chromosome 15 may cause Prader-Willi syndrome? The correct answer is option number 2. Okay, so option number 2 is correct. So here you can able to find patients with that particular syndrome will be having, okay, they have described who don't have any cytogenic deletion, but instead those patients with that particular syndrome will be having 2 copy of this particular region that will be inherited only from the mother. 2 copies, we all know 1 copy will be coming from mother and another from father, but in this particular condition it will be inherited only from mother, it will not be inherited from father. So this unusual form of inheritance is called as maternal uniparental disomy. Okay, so correct answer is option number 2. I'm agreeing with NTA because NTA answer, because many are telling option number 3, but it is due to imprinting of locus in paternal copy. Okay, but here they have given maternal copy. Okay, so option 2 is correct. Okay, next is regarding to this particular statement, like which of the following is incorrect statement, like they are asking. So first thing is regarding to a change of DNA sequence from AGC to ATG non-coding strand will have a major impact. Of course, it is a correct statement, but they are asking about incorrect statement. Second B option, suppressor mutation restore the original phenotype only when the second mutation occurs in original site. But that's not this is a correct statement, like option B is incorrect. Okay, because there are 2 types of suppressor mutation, intragenic and intergenic. Okay, here they just mentioned sometimes second mutation will be occurring in some other location. Okay, next, mutation rates remain same in all organisms, absolutely incorrect. So option B and option C are correct. So option A, option D and option E are absolutely correct statement. Only option B and option C are incorrect statement. So option number 2 is correct for this particular question. Next, 703610. So you can able to follow this particular thing, like option A is correct, option B is correct, option C is also correct. But if you take sorry friends, they are asking about incorrect statement. Okay, for which if you take option A, sorry, I just collapsed. Option A, hemoglobin are most common and widespread respiratory pigment in vertebrate and invertebrate, but hemoglobins are present only in vertebrate. It is not being present in invertebrate. Second is heme structure in hemoglobin varies widely, and different molecular form of hemoglobin, the globin like that they are giving, but option B is incorrect because we all know fetal hemoglobin and adult hemoglobin where globin genes will be changing on. So B statement is incorrect, and if you take statement number C is absolutely correct. So hemocyanin, okay, hemocyanin, they will be having a copper that will be binds to oxygen. So when they are oxygenated, they will be bright blue in color. So we need to omit statement number C because they are asking to give incorrect statement. So statement number D, okay, it is also incorrect because this chlorocurions, they are similar to hemoglobin, but here they are telling they are similar to hemocyanin. Next is option E is correct friends, but here they are asking about incorrect statement. So option A is incorrect, option C is incorrect, and option D is incorrect. Okay, next is question ID 703632. They had given option A and option D are correct, but I do have some confusion with this particular question, so we need to wait for the official thing. Next question is 703581, for which they just mentioned option 2 alone is correct. According to me, option 2 and option 3 is also correct. Okay, so those students who are given either option 2 or those students who are given option 3 will be getting mark. Next is question ID 703605, for which they had mentioned option number 3 is correct, which means they are asked us to pick up the correct statement. So first is transzeatin and isopeptine, but it is isopentenyl adenine and dihydrozeatin are the most common cytokine, but here they had given isopeptine, somewhat different thing. I don't know whether it is a printing mistake, but A is absolutely incorrect statement. So I think those students who are attempted this question will be getting marked. Okay, so I'm not sure about CSIR. We need to wait for the official thing from CSIR. I'm just predicting, I'm just telling my thing. Okay, second so option number B is that CK are present as nucleoside and glycosidic conjugate, but not as nucleotide. But here you can able to find that cytokines are present in plants as riboside, ribotide, and glycoside. Okay, next is a dephosphorylation and deribosylation steps are involved in two steps. This is a correct statement. And lonely guy. Okay, so here if you refer figure number 15.21 in textbook 6th edition, you can able to find it is involved. So according to me, option C and option D alone is correct. So if you have challenged this question, please do tell me your reason, and if you are already having any references means please do tell me. Some students challenged this particular question and some have challenged this particular question ID. And regarding to question ID 703569, according to NTA they just mentioned option 4 is correct, but if you check in this particular article, they have clearly mentioned option 4 is incorrect. Okay, they are asking which parameter of healthy leaf play a major role to reflect the near infrared region. This here you can able to find plant with more chlorophyll will be reflecting more near infrared region than unhealthy plant. So according to me, option 2 is correct. Next is some are telling some had challenged this particular question. So from NTA they just mentioned option 2 is correct statement because they are asking which of the following is not a characteristic feature of aquaporin. Option number 2, they are absent in bacteria. Of course, it is not a characteristic feature of aquaporin. At the same time, option 4 is also correct because this highly conserved NPA, that is asparagine proline alanine, is not present in C terminal. But here they just mention it is present in C terminal like that. Since they are asking not a characteristic feature of aquaporin, of course option number 4 is also not a characteristic feature of aquaporin, because in aquaporin the highly conserved NPA will be located only in N terminal. But here they just mention it is C terminal, which is ultimately not a characteristic feature of aquaporin. So some have challenged this particular question. So according to me, I am agreeing with the NTA because option B is incorrect, because if you BMP inhibitor will be allowing the ectoderm to form neural ectoderm, but here they just mention epidermis. Of course, option D is also incorrect because it is vegetal blastomere. It's not ventral blastomere. Next, option number question ID 703533. This is a correct statement. I just referred various article. If you want, I'll be sharing. Option 3 is correct. Next is 703598. So this is correct statement. I don't know why you had challenged. In the Gilbert book, they had given recent Gilbert book you can able to find. So for this particular question, option A and option B alone is correct because FGF497 from AER, they will be activating the sonic H1 signaling. At the same time, repressive germinal signaling will be repressing the AER. So here you can able to find. Next, some student are challenged question ID 703559. Next, this is in viral question 703538, for which Hydra will be it will be following stem cell mediator, morphallaxis and epimorphosis. If you check previous edition, like if you check edition number 9th edition and all, they will be telling Hydra will be following only morpholytic, but recent research in edition number 12, they just mentioned all the 3 type of regeneration are possible in Hydra. So that's why I had taken thing whenever I'm preparing for any examination, I need to go through the recent edition. Okay, if you're studying at the same time, if you're going for preparing for CSA December, just download the recent edition of the Gilbert book. Okay, and study from that. Okay, and yeah. So very soon I'll be uploading various things regarding to this particular CSA June 2023. At the same time, we'll also be discussing various things. Thank you friends. Thanks for watching this video.